All right, so let's get started. I thought so I have a long time today. We have 2 hours together. So I thought we'd do a little bit of an introduction to sort of the concepts about evo-devo and the shapes of early embryos, and then use some of the work that we are doing in the lab now to illustrate some of these concepts and the things that we are doing right now. I would really like you to interrupt me and ask questions. I have several lines that this talk can go into, so we can make it a choose your own adventure kind of talk according to the things that you find most interesting. But since somebody mentioned that, since somebody mentioned that they never heard about evo-devo before, I thought I start like very general and almost philosophical, right? So one of the most fascinating things about life on Earth is that it comes in all this wide variety of forms, right? We have unicellular organisms, multicellular, things that have branches, tentacles, limbs, fins, wings, right? So this enormous diversity has fascinated humankind for a very long time. And one of the things that biologists that are interested in this ask themselves is like, how are all these different life forms made, right? And there are many different ways in which you can approach this question and try to answer it. The evolutionary biologist, right, focuses on genomes and genetic sequences, and in a way sees these different forms as an expression of genetic information. Like if you want, a life form can be a function of the genome. And by looking at genomic sequences of organisms that share similar structures and comparing them with organisms that don't have those structures, the idea is sort of like, in a very simplistic way, that you can identify what is the code for that particular structure. Like you can compare all things that have wings with things that don't have wings, and then you try to identify what part of the genome is coding for wings, right? So in very general terms. What the developmental biologist does is that they take advantage of the fact that life starts out in a very simple form. At least for many animals, this is a zygote that has a somehow spherical form. And then it's through the process of embryonic development that the more complicated adult forms are made, right? And so in a way, this is the process by which the genetic code is used to make the form. And by comparing this process between many different species, we try to understand what are the principles, like the fundamental laws of development of this process that cannot be altered and always apply, and what are the parts of this process that can be tweaked in order to make different forms, right? And so historically speaking, the aspects of development that we compare or that tend to be compared depend very much on how exactly we are thinking about the process of development in a particular historical setting, right? So in scientific setting. So I want to bring you through how I think about development, and I think many of you will share this vision, which is sort of have been evolving in the past 30 years or so. So what happens in development of many animals is that you start with the zygote, as Akanshi was explaining yesterday very nicely. The zygote undergoes cleavage, which is a series of reductive cell divisions. And so cells become smaller and smaller. So you have at some point a mass of cells. And then these cells start organizing in 3D shapes at the same time in which they start acquiring different functions, right? And so the way that we think about this process is that there is some signal, and this can be a maternal determinant, a protein or an mRNA that is inherited by some cells and not by others. Or there's a molecular signal that is expressed like a morphogen by a group of cells and received by others. But basically, there are differences in the molecular signals that different sets of cells receive within the embryo. And this starts a cascade of gene expression and genitory network programs that alters the functionality, the function of these cells, so that they differentiate in different types. As part of this program, there are changes in the physical properties of cells, like stiffness, cell adhesion, and behavior, like apical constrictions. And so these differences in physical properties are what drive the morphogenetic events that we see during development, so tissues changing shapes. Yep.
Do you think, oh, you just showed it.
I am going to that, yeah, right? So now we know, and I mean, some people thought about this in this way for a very long time, but I want to say in the last 30 years, this has become more and more widely accepted that this is not a unidirectional process, but there is a lot of feedback between morphogenesis and differentiation. And yesterday, I kind of introduced some of the ways in which tissues change shape during development. And you can think about the fact that each of those scenarios, there are ways in which gene expression, cell-cell signaling is changed or altered by these morphogenetic events, right? And just to think about like three ways, if you think about 3D packing or the positions of cells with respect to one another, if there is a cell that is expressing a morphogen and another one that is receiving it, if the tissue bends, they're going to be more distant or closer to one another. And so the signaling events are going to change, right? The signaling that cell experiences is changing. And then there are many examples in which the being exposed to physical forces changes the gene expression pattern of a cell. So if you stretch a cell, there are molecular pathways of mechanosensation that can be activated just because the cell is under stretch. Or there is ways in which these forces can alter the downstream of molecular signaling, that is, the downstream parts of the molecular signaling that is activated by morphogens, right? So we now kind of think about development in this sort of feedback, constant feedback between cell differentiation and morphogenesis. And the way I very simplistically have it in my head is that there is this constant iterative process by which there are some changing in molecular signaling in the genitouring network, morphogenesis happens, and then that signals back to cell differentiation, right? And so you can think about development as a constant feedback that changes over time the shape and the function of your tissues, right? And so in this view, when you want to think about how this process is changing during evolution, right? Whatever change you make in any part of the circle will have effects to the whole thing, right? And so which part of this we focus on depends very much on our own inclinations and what we find most interesting about this process. So there's lots of people that focus on differences in molecular signaling and genitouring networks among species by comparing where certain signals are expressed and what are the differences in the genitouring networks that they activate in different species. And then there are people that are more interested in the changes in physical properties and morphogenetic events and how those feed back into that, right? So I sit somewhere here, and these are the things that I generally start looking at, but I am very interested in how variation in physical properties of cells and morphogenetic events change also molecular signaling, genitouring network, and cell differentiation. All right, so this is what we are going to be talking about. And I will argue that you can analyze this almost at any point in time during development. You'll find some interesting variation. And this begins very early in development. And so during this week, we have seen beautiful talks about a little bit later stages, gastrulation and somite formation and those things. And today I want to tell you a little bit about very early embryos and how they are varying across animal species and how we study those. So what do I mean with early embryo? I mean everything between the zygote and a blastula, so before gastrulation. And what many embryos do in this process is simply to divide their zygote into smaller cells, and then they start organizing in the first more specific shapes that can be different from species to species, right? And so I just want to first bring you through a little bit of showcasing the variety of these early embryo shapes, just so that we are on the same page and we have an understanding of what these embryos can look like. And we're going to start from some of the most famous embryos out there. So this you have seen already. This is a zebrafish embryo that is developing, and the embryonic cells are sitting on top of a yolk mass. So you see these cleavages create this mass of cells that is sitting on one side of the yolk, right? So that's a very specific shape. This one also you have seen already. This is a Drosophila embryo, right? And the shape of the egg is very different, but cells are doing somewhat a similar thing. They are dividing and cellularizing on the surface of the yolk, and in the middle there is a mass of yolk that is not part of the tissue, right? This is anybody wants to guess? Frog. This is a frog embryo, right? We have seen it in a slide, I think, yesterday. Say it again? Dorsal view of the frog, exactly. And so what the frog does is that it has a really nice big yolky embryo, but instead of doing like the fish that it divides the cells only on top, it divides the whole thing, right? And so the whole of the yolk is divided into the daughter cells that organize in a cluster. This, who knows what this is? Yeah. No, they are. So the yolk is being cut, right, entirely. Then there are some differences in cell size. So the divisions are not all equal, and so the cells at the top become smaller. And then eventually there is fluid accumulation, and there is a blastula that is biased. It's not everywhere, but it's on the animal pole, right? So the organization is then you have tissue on top and on the bottom of the blastula, but initially they are all together.
The embryo is just what's on top.
The whole thing becomes the embryo, right? So the cells at the vegetal pole are yolk rich, so they are going to be used in a different way, but the whole, all of the cells become. Yeah.
So the information of the geometry of the egg, where is it? Has that evolved also? What controls that?
The shape of the egg and how that is controlled is a very good question. It is definitely controlled in some way. It evolves and it changes. And I don't know that we know exactly what is it that makes eggs of different shape, but obviously it's during oogenesis and how those are created during in the ovaries, right? That is a very good question. Also, there are groups in which there is a humongous variation in egg shapes, like insects have like all kinds of oval and comma-shaped kind of eggs, and there are other species in which the initial shape is kind of similar. It's spherical, right? The nematodes also. What is that one? Do you know? C. elegans, exactly. The nematodes also have variation, so the eggs are oval, not spherical, and like how much is oval and less.
So, but this information, is it in the zygote or is it additional information?
Well, if you want, it's in the egg, right? So it's before.
On top of what's in the zygote.
The zygote is formed once the egg and the sperm form together. So if you want, it is an additional information. Yeah, it's not the gene. Well, it is the genome in a way because the mother is making the egg, right? So there is information in the genome that allows you to make an egg of a certain shape, but it's not in the zygotic genome that is going to create in a way. It is changing so there is a lot of effect of maternally deposited protein and shape, which are really important for the very early phases of development, right? So I sometimes like to think about the fact that if you take a genome and you put it in an Eppendorf tube with water, it's not going to do anything, right? So you have an initial set of conditions when you start development, which is your genome plus all of the stuff that is in the egg when you begin. So in that sense, yeah. Yeah.
Is there any link between the shape and whether it's external or internal fertilization? Like whenever it's internal, is it mostly spherical? When you have to lay, then there's some sort of...
No, you think, no, just, I mean, on top of my head, I can think of examples of eggs that are laid or kept or fertilized internally that have different shapes. So not necessarily. Yeah.
How complex is the yolk in terms of signaling or mechanical properties?
Very, yeah, we could talk about that for an hour, but yeah, so there is, the yolk proteins are nutrients, but they also change, obviously there is some mechanics here, right? So if you look about this fish embryo, right? The yolk is on the bottom, you see this flows, right? So that is basically the cytoplasm, the liquid part of the yolk that is being pulled up as the embryo divides, right? And so there's definitely differences in material between the more liquid parts of the cytosol and the yolk granules that are like very dense. How this changes across species, it's a very interesting question. There is some data out there that we can go look for together, but it's been studied in not a lot of species. Like there's something very interesting, for instance, in mollusks that have very, very granular yolk granules, like very stiff, Like they look like pebbles. And how that changes the dynamics of their cell division is actually not clear at all. All right, so C. elegans also does whole, the whole of the egg is divided into the cells, right? But then the egg is a different shape, and it is absolutely dependent on this coat that it has around. So if you take these C. elegans embryos out of its envelope there, it's not going to develop in the same shape. The cells are just going to be linear, like they're just going to unfold, right? So there is differences in how much embryos depend on their embryonic coats. This is how they're called in general. What is this? Does anybody know? This is much less famous of an embryo. But beautiful, isn't it beautiful? So this is a marine embryo. This is a sea star embryo that is developing there. And so as you can see, it does a similar thing as the C. elegans and the frog, right? So it does all of the yolk is divided into their cells, but then the cells organize very quickly into this monolayered epithelium that has a cavity, like a large cavity inside, and that's the blastra, right? So similar concept used in a different way. And then finally, I want to show you this one. This is one of the very few videos of a spiralian embryo that is developing. This is a marine snail, a slipper snail, Crepidula fornicata. And this also is like the cell divisions encompass the whole of the egg. So it's split, but then it does these very asymmetric cell divisions. And so you have these very large cells at the vegetal pole that are more yolk rich, and these tiny cells on the animal pole that are more cytoplasmic rich. And then on top of that, it does this weird thing that each cell division makes the cells position themselves in a little bit of a spiral. And so that's why this is called spiral cleavage. So in the end, you end up with an embryo that has a big mass on the bottom and a mass of smaller cells on the top that looks kind of like a zebrafish embryo, but the yolk is not separate from the cells. It's divided into the vegetal bigger cells, right?
The spiral shows
So this is interesting. The directionality of the spiral cleavage predicts the directionality of the coiling of the shell. Nobody knows why and how, but it is known that it is dependent on acting binding proteins. Specifically, there is one species of snails in which this has been shown to be dependent on DAM1, and the allele that the DAM1 is sets the directionality of the spiral cleavage and then the directionality of the coiling of the shells. So one of the things that my postdoc advisor is doing is that she's looking at the generative networks of shell formation during development to try and figure out how these shells are formed, which is not clear at all. You know, how do you make this material of a shell, which is based on calcium, but it's very resistant, right? It's not brittle at all. How do animals do that, and how do you get the variety of shell structures and materials? So there also we can talk about for hours. You want to see one more weird thing? I'm gonna say yes if this works. Let's see. Yes, right. I have to, sorry, I think I have to stop sharing. I'm sharing a different window. Here. Can people on Zoom see this? Okay,
yes.
so, okay, perfect. This is the weirdest thing, right? This is another marine embryo. Look at how weird the shape is. Like you think, what can you make with just a handful of cells? This thing is making a U shape, right? And it has these big cells on the top, on the bottom, and tiny cells on the top. And then this is when it starts gastrulating. This is a ctenophore, right? So comb jelly, if you have ever heard of that. So we have like an enormous variety of shape and forms and dynamics of early embryo development that we can sample to try and understand what are the principles of shape formation and shape variation during evolution. Yep.
So is the gradual loss of synchrony always correlated with the number of divisions across?
Mm-hmm. Okay, so let me see if I can start this again from there. I'm gonna get there in a second. All right, so short answer is no. There is loss of synchronicity is very different in different species. For instance, the sea star has very synchronous cell division until the 512 cell stage, which is where we think then, right? And then it starts being asynchronous. Sea urchin starts having asynchronous cell divisions already at the 16 cell stage. And that's because it does, instead of making mostly symmetric cell divisions, the sea urchin does a very asymmetric cell division at that point, and it creates these tiny cells on the vegetal side. And those are much slower at dividing than the other ones.
Somehow connected with.
It is very connected to asymmetry, not always also. So there's a little bit of variation there. But generally, when cells are smaller, they start having longer cell cycles. And before that happens, the cell cycle of the cleavage is actually very unusual because it only has S and M phases. So there's nothing to do. So it's very fast, and we just divide and divide and divide, right? It is indeed, yeah. So when they start being asynchronous, then it's also generally when they start using the zygotic genome. And interestingly enough, the sea urchin is one of the examples where you start using zygotic genome already even earlier, like at the 2 cell stage, there are some zygotic genes that are expressed. For other species, and a lot of species out there, I don't really have that information, but it's generally that is when it happens. Yep.
Just on the snail. So
Mm-hmm.
in the nature, when you look for a chirality of the
Mm-hmm.
do you see 50-50?
It depends on the species. So there are species that are just one-sided, and there are species that have populations of left and right coiling. So there is a lot of variation. I will say, like, the majority of species are obligatory chirality, either left or right, but there are a few out there that you can find shells of snails with both.
The one nice thing is in the earliest part of their division development, they might all look very similar. So have people looked at the actin and other things localized in this species too, which has only one particular chirality as opposed to different fragments and so on and compare?
I think probably, so there's this whole body of work on this one species that has both, and they have identified this DAM1 actin binding protein that is necessary for that drives that difference. And now on top of my head, I cannot remember if they looked at other species and what these alleles look like in other species as well, but we can go look it up. All right, so. Later on, not at the cleavage stages though. Yeah.
So it's downstream from the.
I think so, yeah. All right, so what are the determinants of this variation of shape? So some things we already touched on, and the most obvious, it's obviously the starting shape, right? So you can have a perfectly spherical egg and a not so spherical egg, and so that will change your starting condition. Then the other one we have seen is the type of cleavage, right? So we've seen zebrafish and also chicken, right? Do this thing that they have a very large yolk, and then only the cells on top of the yolk divide. Initially, they are not even entirely separated from the yolk, but the cell division only occur on one area of the embryo, which is the cytoplasmic one, and those are called meroblastic cleavages, right? And so you can imagine how changing the ratio between the amount of yolk and the amount of cells that you have is going to change the shapes. So you can think of a chick embryo as a very enlarged yolk of a zebrafish, and then what's going to happen is that the cells are going to flatten down, right? So very simplistically speaking, you can think about that relationship and the variation thereof as a way to generate more diversity in the early embryos. The other type of cleavage that we have seen is holoblastic, which is the ones that in which the whole of the egg is cleaved and separated into cells, right? And so this sort of thing creates variation. Even if you just compare embryos that do holoblastic cleavage, right? The way that the cell divisions are patterned is gonna affect the shape of the embryo, right? So this is one of the large determinants of early embryonic shape, is like in which direction are the cells dividing, right? And these are examples. This is a picture that I took from this very nice review from Nicola Mink. If you're interested in the role of cell division in early shape variation, there are two groups that have done like really extensive work in comparison of these patterns across species. One is Mink, and the other is Isaac Salazar Ciudad. They have several really cool papers on this. But in general, right? So if you have a cell that divides in the tangential orientation, like the one on top, you're gonna expand a layer. If you divide in the other direction, you might make two layers, right? So obviously the direction of the cell divisions are gonna be important for determining the shape of your early embryo. The other thing that we kind of touched upon is symmetry versus asymmetry of cell division. So you can divide a cell into equally or more or less equally sized cells, or you can divide a cell into one that is much tinier and one that is much larger, and that is gonna change the shape, right? Not only the shape, but also the synchrony, right? So I haven't put that down here, but also synchrony and asynchrony of cell division changes the shape. Because if all the cells are dividing at the same time, they are undergoing this rounding and changing their properties all at the same time. And that creates a certain shape. If half of the cells are dividing and the other half are not, they can compensate and organize differently. We'll see some examples of this. And then the other aspect is
heteroblastic versus holoblastic, is it associated with embryo size in the end or not really?
Not really. Like the frog, there are frogs that are, so the xenopus is 1 millimeter big. There are frogs that have eggs up to 6 millimeters big, right? What has been seen is that with larger and larger sizes in the frogs, what happens is that the cleavage becomes less stereotypical and less predictable. And so there's an idea that perhaps there is a size limit in which cell division, that allows cell division to be really reproducible and robust. And if you go over that size limit, you might end up with still a holoblastic cleavage, but it's not as beautiful and regular as the one that you see in the xenopus. But so there's that.
What's interesting also is that you can have this asymmetric cleavage like in piranhas, and then land on bilateral symmetry.
Right.
So when you acquire the bilateral symmetry, not necessarily from the beginning, it's
Oh, I see what you mean. Well, I'm not sure that I never thought about it that way. I always thought, even the sporanian embryo divides, the first division divides in two. In some species, those are already predictive on left and right, right? So how the bilateral symmetry relates to the symmetry of cleavage, it's an interesting question. I never thought about it that way, but
in front.
right. Yeah, no, I've looked a little bit into this for the urchins and the sea stars because I was doing these lineage tracing experiments to see if the early cleavage predicts. And that is also species specific. Like in some species, the first cleavage is already predictive of the left and right symmetry. In some species is not. But does that mean that the embryo is not bilateral yet? I don't know, yeah. Any other questions on this? Okay, so the other determinant of this early shape is obviously like how packed together the cells are, right? And you've seen sort of in these pictures, there are big differences, both in the initial packing and then how this packing evolves. So some embryos look very compact from the beginning. Some embryos look very loose, right? And so a framework to kind of compare and think about this compaction, it would be really useful, right? Something that you can use across different species and across different organisms. And as Akanshi was saying yesterday, what are the things that determine how compact a group of cells is? It is how adhesive they are to one another, right? So how much they like to stick to each other, how much they don't like to be in the medium they're in. So if they really don't like that, they will compact a lot more and they will try to minimize the surface to make a nice sphere, right? Because that's the shape that minimizes the surface to the outside. And so, And so there's been like a really long history of different types of work, try to find a framework to describe and model these differences in compaction with relation to shape, but also with relation to cell sorting, right? I'm not going to go into the history of this, but it started with the differential adhesion hypothesis, where the idea was that if there are two populations of cells that have differential adhesion to one another, so let's say that you have a cell type A and cell type B, and you mix them together, but cell type A likes to adhere to one another much more than they like to adhere to B, they will eventually sort because of this differential adhesion preferences. This has been then expanded to think about differential interfacial tension. So not just about the adhesion between cells, but also about thinking about how they interact, their surface interacts with medium and other interfaces, right? But all of them sort of are based on the principle that you can think of cells as liquid droplets. And so that their positioning with respect to one another is mainly governed by their surface properties, right? And so this is something that Jean-Léon Maître and Hervé Toulière have used as a way to describe the shape of embryonic cells in zebrafish, in mouse, and human. And it's been very useful to model how compaction changes and evolves in these embryos, right? And basically what the major point of this is the first equation of two equations that I have in this talk is the Yang-Dupré equation, which is just saying that the angle that is formed by two cells in contact depends on the balance of tensions that are occurring at that point of contact, right? And so by measuring the angles, which are a measure of compaction, and measuring the surface tensions, you can get an understanding of what are the parameters that cause those particular type of cells to compact or not compact, right? And so Jean-Léon and Hervé have used this to describe really nicely and model really nicely the compaction of mouse and human embryos. And this is a particular case of an early embryo. Kangxi showed some video yesterday, so I didn't put them back in. But what they do is holoblastic cleavage, right? And then all the cells are together, packed together, but they start kind of loose, like in that picture. And then as development progresses, the first thing they do is that they become more compact. So they minimize the surface to the outside, and they become this tiny little ball of compact cells. And so they have modeled this as a change. What they have found is that the parameter that changes most is the surface tension to the outside, which is controlled by the cortical tension of the cells, right? And so the mouse embryo cells increase their surface tension to the outside, and that causes them to compact, put very simply. So this, I think it's a very nice framework to try compare how shape of embryos are changing depending on the properties of the cells. And it has been used here very nicely. The thing is that when you look at variation of shape among many other species, the situation can be a little bit more complex than that, right? So you have the cells, they might not be entirely packed together. So you have medium also in the middle of the embryo. And then often enough, if not almost always, embryos are encased by a coat. And the relationship between the cells and the coat, how they interact with each other physically, can change things and complicate the situation a little bit, right? So I am now about done with this. Yeah.
Just I notice in these images, the cells all look like they have equal pressures.
Mm-hmm.
Are you going to talk at all about other mechanical aspects?
Yeah, I was not going to go into the pressure, but it's obviously important. Yeah, but so you can have hydrostatic pressure that keeps them and gives them different properties. In this framework, that would result also in a difference of surface tension, basically. So if you are very pressurized, it's going to be more difficult to deform your surface. And so effectively, it's going to go change the surface tension. And so I know, like Jean-Léon and Hervé, they kind of focused on the cortical tension as the major effector of that, but it doesn't have to be, right?
So can you essentially use those two parameters per cell to describe also essentially? And in these particular examples, the fact that you see very simple symmetries is because it's just assumed that all the cells are
the same.
Are symmetrical.
Yeah.
But it would make a huge difference whether you have a shed around.
Exactly.
Or not.
Yeah.
Because in mouse you don't. So
Exactly. Well, in mouse you do. You can remove it, but you do have the shell, right? You have the zona pellucida. You can remove it, and then the embryo does just fine.
it compacts the same way. So
It compacts this. It can compact.
I suppose here you would just increase the osmolarity, or decrease the osmolarity, and the cells would.
Right, and they can.
They fill the space.
Yes, in some embryos that happens, in some other embryos it doesn't. In the mouse, so what Jean-Léon and Hervé, and then also Edouard Anezo had a nice paper on variation of compaction in mammalian embryos. And so, grossly speaking, the compaction happens even without the coat. But then if you do have the coat, the movement of cells within that coat is different. And it seems to be more efficient if you have a coat. So it kind of limits the rearrangements that the cells can have. So, yeah.
What about the stimulation of the blastomeres? Does that change, or how does that play into the tension on the surface?
The what?
Sometimes the ciliation, sometimes.
Oh yeah. Yeah, I am not sure. So all of this happens before. So when you say cilia, I am thinking about the fact that most of these animals, at some point, each cell is going to develop a big cilium, and that's what they use to swim. I'm not sure if that's what you are referring to.
Is at the blastula stage.
Right, that they have cilia and phylopodia. Yeah. So that is not considered here, and I think the idea is that it's also gonna end up changing the overall surface property, and so these are kind of mesoscopic property of the surface. And then the question of what exactly is setting that particular surface tension in that particular cell, it's a whole other set of questions that one can then go and ask species by species and try to understand how those parameters are affecting the overall, the effective, I call it effective surface tension, right?
Yes, can I ask, so if you put the cell in isolation, does it try to round up or reach a particular perimeter? Or
okay, so we are starting
biologically
the adventure. Let me see if I can. That again depends very much on the embryo, and I just have this beautiful movie, so I'm gonna try to pull them up. This is a new. This is kind of a new project that we are working on. It's a very old finding and a new project for us. So, let me see. Share. Okay. Okay, people on Zoom can see this? I'm gonna assume
yes.
Okay, so I'm gonna start this again. All right, so this is very, very fresh stuff. So one thing that you can do with the sea stars and with the sea urchins, right? You can chop them at the 2-cell stage, at the 4-cell stage, and isolate the blastomeres. And then these blastomeres will make tiny, almost perfectly scaled down larvae, right? Embryos. And so what happens when you take them out of the envelope? So the first one is a wild type embryo. The second one is an embryo that is being taken out of its envelope, which you need to do in order to separate the blastomeres. And then you have a half and a quarter, right? And so if you look at the half and the quarter, it sort of answers your question. These embryos start there, look at that. They kind of make some sort of shape, and that shape depends on where you put them. So they tend to flatten out. And then at some point, they really want to close. They really want to make a sphere. And so that's another concept about these early stages of development. The properties of these cells are changing quite rapidly in comparison with the timescale of these morphogenetic events. And so I think the thought that we have right now is that the moment in which they really want to change is probably when they are becoming more epithelial, and they really don't like to be splattered outside in the seawater anymore. There's a recent paper in a sea urchin species that does like a completely flat layer of cells until at some point they cup up and close and make a blastula. So,
Vanessa?
Yep.
Sorry, the one in which the coat is removed, are there more cells or is it less compact?
It's That's a good question. I'm not sure how to the cell density. If you look at the number of cells per tissue, it's even once it closes, it's even more compact. But what's happening is basically there is no envelope to keep them nice and spherical anymore. So they're kind of relaxing. And so it seems bigger because some of the cells that normally would be on top are now falling on the side. And so it looks bigger. It's less.
So they just have more surface area. They're not.
Exactly.
They haven't minimized. Okay.
Exactly.
Yeah.
And then eventually they close and then they start pressurizing and they become a beautiful sphere again. Does that answer the question?
Yeah.
More or less?
Do you know if the smaller embryos, so they have smaller cells or they have less cells?
They have the size of the cells is equivalent to the size that they would have if they had stayed in the whole embryo, right?
So they have less cells. So
they have less cells and they really become smaller. So the half embryo will develop into a larva that is exactly half the size of a wild type. The cell size remaining variant, at least for the cleavage stages. We haven't checked at the end, like at the. Yeah, it's really, yeah. I am really excited about this. Okay, back to the original adventure. Where is it? Here. Okay. Zoom. Share. Here. All right. Okay, so then, there is this concept like still the shape of these early embryos is really important for the reasons that we talked about before, right? Because the shape sort of sets which cells are signaling to which. And for each species, you have to have a certain shape of the early embryo in order for development to be successful, right? So we have sort of this conundrum, which is, oh yeah, I want to tell you about this. This is a good example of that. This is the Ascidian embryo. And this is work from Patrick Lemaire group, but there's also work from Claire Hudson and Momosen, McDougall and Remy Demlard that have looked at this. And what the Ascidian, so what they have done, what the Lemaire group has done in this particular paper is that they looked really carefully at the 3D cell shape and 3D organization of the early embryo cells in 10 different embryos. And they have looked at the variation between these shapes. And the Ascidians are a bit of a particular group because they are really, really invariant. So species that are very, very far away from each other evolutionarily have the exact same shape of an early embryo, and you can recognize the same cells, right? And so in this paper, they took 10 embryos of the same species, and they looked at how variable at the level of area of cell-cell contacts between cells that is from embryo to embryo. And they found that it's really not changing almost at all. And so this creates a situation in which, and so they hypothesized that this is because the exact position of cells is really important for cell fate determination. So there are several signaling events that depend on which cells are close to each other and how large is their cell-cell contact area. So in the sense that there are some events in which if you don't have a cell-cell contact area of a certain amount, above a certain threshold, the signaling is not going to be effective and your cell fate specification is going to be messed up, right? So we have this situation in which the shape and the relative position of cells in the early embryo, it's really important. But then also we have this huge variation of early embryo shapes, right? And so this is always a bit of conundrum that I have thinking about evolution. There has to be some variation for evolution to work, right? But still, you do need a particular shape for each species for development to be successful. So the questions I have and that we are trying to very generally address are of this type, right? Like how does the shape of early embryo vary? How much? What are the main determinants of this shape variation? And what are the effects of this variation onto the rest of development and cell differentiation and so on? And so we have a few minutes, so let me see. I think I'm just gonna introduce you to the model systems that we use, right? And as Akanshi mentioned, I did my PhD in zebrafish in Carl Philipp Eisenberg lab, where you can really nicely look at the shape of cells and the embryos and the interactions between cells. And we have amazing tools for cell-fate differentiation, genetics, and everything. Evolutionary comparisons were a bit difficult, right? Because fish need a lot of care. And so at the end of my PhD, I sort of explored what other systems can we use to study these sort of questions that have not been exploited that much in this particular respect. And I took the embryology course at the MBL, and I stumbled upon, I very naively discovered that people were working on these systems that are echinoderms. And echinoderms are a very large family of marine animals that come in all kinds of different forms as adults, right? So you are probably familiar with sea stars and sea urchins, but we have sea cucumbers, brittle stars, feather stars, right? And so this variation of shape and forms is incredible at the adult stage. They also have a larval stage, which is what the embryonic development results into for many species in each group, that has a specific shape depending on the group, right? And so this is just a schematic of iconic or particular larva shape of each of the classes. And they're very beautiful. In many species, they develop in just 2 or 3 days. So they are experimentally very tractable. And there are many characteristics that you can sort of find in all of the groups, right? So if this is a sea star larva, you see it's bilateral, right? And then the pentaradial comes at metamorphosis. So that's interesting. They're all bilateral. They have a mouth, they have an anus, and then they have these things here that are called ciliary bands that are the structures that they use to swim around. So these are planktonic larvae. They go around in the ocean for a few weeks, eating up until they are ready to metamorphose, a bit like insects, right? You look at that sea urchin embryo, it has a bilateral symmetry, mouth, anus, ciliary band is going all around. This guy also has a skeleton in the larva, which the sea star doesn't have. The sea cucumber has a tiny skeleton, right? So you can appreciate that there are things that stay the same through evolution and things that change in a very particular and interesting way. And so they have been modeled for evo vivo for a long time. And we have a really good handle on the genotary networks and the variation in the genome that causes certain things to stay the same and certain things to change. So to me, this is like this seemed like a really good system to start looking at this variation in early embryo shapes, especially when I looked at the early embryo shapes, right, which has some very interesting variations. Some of this we have touched upon already, but this is a sea urchin embryo, just to give you an idea of what I'm talking about. You see how it starts. It has its zygote, its coat. It does this holoblastic cleavage. The cells are pretty nice and compact. And then the first thing that it does is that it organizes in this blastula. This is a monolayer epithelium that encircles a cavity. The cavity is pressurized, right? Okay. Sea star embryos does a very similar thing. Starts as a zygote in its coat, which is called a fertilization envelope, ends up as a monolayered epithelium that encircles the cavity, a blastula. Blastula is pressurized, does holoblastic cleavage, but you see how the cells are very differently packed, right? So the cells are very loose. There are between the cells at the beginning where fluid can go through, and then they kind of organize on the inner surface of the envelope until they have closed the cavity, right? And what you can do is that you can do really, they're very accessible. They develop in seawater. They are transparent. And so you can do this imaging quite easily, honestly. You just put them on a confocal, and you can image what each cell is doing through this process of blastulation, right? And you can get really quantitative about it. And so what you can see here is that there are these oriented cell divisions, right, that keep the cells on the surface. And it seems like the tissue is sort of thinning and expanding until there is no more space, and then all of it is closed, and you have made a blastula, right? So you can, because they're so transparent and there is just one layer of cells, right? You can do 3D segmentations of this, and you can get an understanding of the shapes of each individual cells, their volumes, which neighbors they have, how many neighbors, how big are the contacts with their neighbors, and you can do this over time as the embryo is developing, right? So I am gonna just stop here for a second. I just wanna tell you a little bit about different imaging methods and what are the limitations and advantages of each of them, right? So this confocal, super easy. I mean, super easy, relatively easy. You have a confocal in almost every lab now, and you put them on a slide and you image them from the bottom, right? And this gives you good images for about, like really good signal for about a third of the embryo. And then you can't see on the other side though, because these embryos are still 200 microns inside, so you cannot see on the other side. But you can image like multiple embryos per night. This process, I didn't mention, but this process happens in about 16 hours tops. So it's an overnight movie, and you can get multiple embryos. And the data that is produced is manageable. And you can do this sort of segmentations and analysis on a good gaming computer, right? But if you want to have the whole thing, you need to go into using different techniques. And so now in collaboration with Sebastian Streichen and Peter, who is a student in Sebastian's lab, we are doing light sheet imaging of the C-star embryos. And this is just, this is very new data. So what you're seeing here is at the end, sorry, at the end, no, come on. Here, like when the blastocyst is already formed, you see this, we have a really good view of all the cells all around the globe. And that allows us to potentially do nice segmentation all over. Like in this example that Sebastian sent me, where we have the early development, and we can get really the shapes of all the cells all around. And then you get this also capability of really doing cross sections in the middle, and you can see how the cells are really just organizing on the outside more and more over time, right? Or
so at this stage, there is an apical-basal polarity, right? But when does it set up?
From the very beginning. At the 2-cell stage, you already have an apical-basal polarity. And the way we know this is that there have been experiments where people have separated the two blastomeres and then flipped one. So if you put them back together in their original orientation, they will make one blasta. If you flip them, they make two separate blasta. So, and there are some, we don't really have good markers for now. We are cloning all of the apical-basal polarity markers to see it, but there is definitely polarity to begin with. And so one of the questions that I find super cool is like, they have the polarity, but they cannot really behave like an epithelium until they have closed everything, right? Because there's fluid flow.
So chiral symmetry breaking is broken very early, but the tissue scale is a little bit unclear.
Chiral symmetry?
Like if you have a polarity, that means that this axis is different than the other axis.
Yes.
So that's basically a sign of chiral symmetry of the single cell.
Yes, absolutely. You also have, the animal and vegetal axis of the embryo, which is telling you what the future anterior and posterior will be. It's already set in the egg. That too, right? So those are two different type of polarity. All right. So we are at the hour, and I think I can stop here, and then we can go to the rest later if there are more questions about what we talked about. Yeah.
When you look at this at the scale of like second or less, then think of these as fluctuating membranes. Like this, going back to the role of shape, if it was flat, it would be hard to maintain the flat shape with fluctuations compared to a sphere.
Yeah.
Like if that, like
possible.
Some embryos when they start from a flat sheet, for instance.
So there are some jellyfish that do that they start as flat and then they close at some point, very much as our embryos look like when we remove them. I personally have not looked at them, but there are papers about it. Very sparse. Like, so one issue with all of these critters is that they're not really laboratory animals. So you have to get them from the wild, hope that they're ripe, get the embryos, do your experiments. So the body of work that is being done on them is not as extensive as with other model systems. We are going back to them. Like there was in the beginning of embryology was all that, right? People at various stations and a lot of marine stations just going to the beach and grabbing stuff and looking at embryos. And that's how the very first, the Drish experiments and everything were done. Then with the molecular biology boom, we kind of went into the details of that in a few embryos. And now with the techniques and the technology that is available with CRISPR and other things, it's expanding again. So I'm hoping there will be more. Yeah, but I think a project where, you know, you get these jellyfish embryos and you look at the variation, you know, you image 100 of them, you look at the variation of shapes and fluctuations and how they, if they are canalized to the blastula at some point, I think that would be super awesome. It's totally doable.